So welcome to Excelsior Academy, guys. And in the last lecture, we have started with the translation, right? So wherein which we have discussed about the mRNA, messenger RNA. Okay. So today also we are just going to talk about the tRNA, like what are its special features, like what are the important points to remember about it. So as we know, see, basics of translation, everyone remembers that we have a mRNA, then tRNA, which brings the amino acids, and then we have got the ribosomes. ये तो हमें पता है, but then we also need to know the special features or the special characteristics of each of these components so that it becomes easy to understand translation better in further classes. Okay. So the tRNAs are the adapters between codons and amino acids. So we know they are the adapters. इसका मतलब क्या है? tRNA are able to read the codons because they have got the anticodons. And on the top, they have got the amino acids. So they are like an adapter between the codons and the amino acids. So there are many types of tRNA molecules, but each is attached to a specific amino acid and each recognizes a particular codon or codons in the mRNA. So as we know that we have got from the genetic code table, we know that we have got 64 codons, correct? Out of which 3 are stop codons, the remaining 61 are there, which is coding for the amino acids, right? And we have only 20 amino acids. So guys, what do you think? Like how many tRNAs do we have in the cytoplasm? Is it 61 tRNA molecules or 20 tRNA molecules? What do you think?
20.
Sure about 20?
Yes, ma'am, 20.
Okay, good. So that is a good guess because see, now why don't we have 61? Because the 20 tRNA molecules are designed in such a way that they can also read the other codons and code for the same amino acids. So those are known as cognate amino acids. So we'll talk about all this in detail. Now, what makes this special features is like how the tRNAs are able to get coded, or they get attached to the amino acid, same amino acid. Like for example, if I say that we have got 3 codons coding for phenylalanine. Okay, so all the tRNAs are having the different codons but getting the same amino acid. ये किसकी बदौलत है? Like why? Who is making this possible? Any idea? So as you said, there are 20 amino acids, so 20 tRNA molecules are there. Okay. But we also know that there are 61 codons. So instead of having 61 tRNA molecules, जो कि economical नहीं होगा for any particular cell, right? So it is always good to have 20 tRNA molecules. But who is it which makes it possible, which is able to read the codons and giving the same amino acid? Yes? Any guesses? Have you heard of tRNA synthetases? Yes, so we have got 2 types of synthetases, class 1 and class 2. Okay, so these are the enzymes. Okay, these are the 2 classes of enzyme which is helping in charging of your tRNA molecules, even though they have got the different codons, but they are charged with the same amino acids. Okay, so such tRNA molecules are also known as iso-accepting tRNA molecules also. Okay, so we'll come to that. Slowly we'll come to that. Okay, so tRNA molecules are between around 75 to 95 ribonucleotides in length. Although the exact sequence varies, all tRNA have got the special features which is common. Now, first thing, which the peculiarity about tRNA molecule is about the terminus, that is 3' terminus at which your amino acid is going to bind. So that is going to remain constant. CCA. ये sequence आपको याद रखना है. The direction also you need to remember. Question में क्या होता है? कभी-कभी they give it like this. बट हम हमेशा कैसे याद करते हैं? CCA. सो उसमें आपका मिस नहीं होना चाहिए. जस्ट गिव इंपॉर्टेंस टू द डायरेक्शन ऑल्सो. सो 5-CCA. तो आपके ए में यहाँ पे आपका एमिनो एसिड जो है चार्ज होगा. ओके. Now, consistent with this absolute conservation, the 3' end of the sequence is the site which is getting attached to the cognate amino acids. We'll see in this today's lecture how the tRNA molecules are getting charged. Now, the second feature of tRNA is the presence of the several unusual bases in their primary structure. Now, if you ask what kind of these bases, like for example, as mentioned, pseudouridine, dihydrouridine present in tRNA molecules, इनका function क्या है? So it has been observed that these are not helping in the codon recognition, but they do help in the function of the tRNA molecule. Okay, so eventually helping in enhancing the translation process. Okay. So these unusual features are created post-transcriptionally by the enzymatic modification of normal bases in the polynucleotide chain. So okay, so this is one of the example where you have come across about the modification. So later on, whenever someone says PTM, okay, PTM usually हमेशा एग्जाम में PTM modifications के बारे में रहता है, but those are all your post ट्रांसलेशन मॉडिफिकेशन, ठीक है, जहाँ पे आपका एसाइलेशन आ सकता है एंड फॉस्फोराइलेशन आ सकता है, ऑल दीज थिंग्स विल कम. बट इफ दिस स्पेसिफिकली मेंशन पोस्ट ट्रांसक्रिप्शनल मॉडिफिकेशन, देन दिस इज वन एग्जांपल वेयर यू कैन talk about. So that is the tRNA molecule having unusual bases. For example, the uridine is getting converted to pseudouridine or dihydrouridine. So these are the examples of post-transcriptional modification. Okay. So for example, pseudouridine is derived from the uridine by an isomerization in which the site of attachment of the uracil base to the ribose is switched. Okay. So आप देख सकते हैं यहाँ पे. So normally कैसे होता है uridine में at N1 position the ribose is attached. But in the case of pseudouridine, it is the 5th position. Can you see the difference? And able to remember, guys? Can you remember? Abhi se yaad karo, like when the lectures are going on, please do remember. Okay. Okay. So I'll just repeat once again. So the uridine, which is having N1 position ribose, it's getting modified and it is going to the 5th position. So this is how your pseudouridine is there. Now coming to the dihydrouridine. So what is happening to the dihydrouridine? You can see it over here. Normally, there is a presence of a double bond at 5 and 6 position. But here, what happens? There is an addition of H. So, dihydrouridine. Rest all is same. Can you see the difference over here in the structure? Yes, Tanvi, Akshaya? Able to follow?
Yes, ma'am.
Okay. Okay, it's very simple. It's just that we need to remember certain topics over here. Now, likewise, dihydrouridine, it is derived from the uridine by the enzymatic reaction where the double bond has been reduced. Now, when you say it has been reduced, so यहाँ पे आपके H bond जो होते हैं add हो जाते हैं. It's the same figure, okay, from the previous slide. Okay. So the other unusual bases which is found in tRNA molecules includes hypoxanthine, thymine, methylguanine, etc. Now, one second, huh? So these modified bases are not essential for tRNA function, and but the cells which lack these modified bases shows reduced rates of growth. यानी कि क्या उसका translation अच्छा नहीं होगा. So this observation suggests that these bases lead to improved tRNA function in the codon recognition. Okay, sorry, I basically said that it is not for the codon, but it is for the codon. Okay, codon recognition. And later we'll see how these are helping in those recognition. Okay. Yeah. Okay. Now, tRNAs, they share secondary structure that resembles a clover leaf structure. So this is how your clover leaf structure it is. Okay. This is very commonly shown everywhere. And this is like your two-dimensional structure. Okay. This is your two-dimensional structure. So this is your pseudouridine loop. This is your dihydrouridine loop. Okay. And this is our acceptor arm. So आप देख सकते हैं sequence आपका 5-CCA3-. यहाँ पे आपका amino acid attach होता है. Okay. So obviously we know whenever a amino acid is attached, a kind of peptide bond should be formed, right? So वहाँ पे आपका condensation reaction होना चाहिए. So obviously here also the OH group is being used of your A. Okay. उसका जो ribose है adenosine का यहाँ पे उसके राइबोज में आपका OH जो है आपका एमिनो एसिड अटैच होगा. Here also the difference comes in the tRNA molecule whether it is the 2-OH or it is 3-OH. So depending on that whether it is 2- or 3-, you have got different class of tRNA synthetases. I repeat. So this is the adenine. Adenine ka ribose, it has got 2-OH and 3-OH. So depending on the tRNA synthetase classes, class 1 and class 2, depending on that, the tRNA molecule will get charged with the amino acid either to the 2-OH bond or to the 3-OH. Okay? Is this part is clear to you guys? Huh? Is it clear? Okay? Huh. So the tRNA share a common secondary features or structure that resembles a chloral leaf. Now, due to the regions of self-complementarity, now if you see the structure of the tRNA, you can see that these dotted lines, what these dotted lines represents, Tanvi, what these dotted line represents?
Some bond between them.
Yes, which bond? Which bond? That is more important. So when you see that it is having a, it is within, see, if you see this is 5- and 3-, this is your RNA only, right? So in the RNA, if you see such kind of structures forming, a loop-like structure is formed. These are nothing but loop only, right? So this is called pseudo-uridine loop. This is your dihydro-uridine loop. Then variable loop is there. All these loops are formed because of the self-complementarity. Now this is only happening only with the presence of the dash bonds. Akshay, would you like to try which kind of bonds are present?
Hydrogen bonds.
Yes, it has to be hydrogen bonds. So when you say self-complementarity, what does it mean? It means that they are going to be complementary to each other. अगर यहाँ A है तो यहाँ U है, C है, ये G है, यहाँ C है, ये G है, A है, U है, A है, U है. So because of these complementarity in the sequence, they form such structures. Okay, so these are nothing but the secondary structures. जो loop like कहीं कहीं पे हम hairpin loop बोलेंगे. यहाँ पे for example they say as a pseudo-uridine and D loop. It's all because of the unusual bases present in this region and in this region. That's why these loops are defined in that way. Okay, so these are your hydrogen bonds. So whenever we talk about RNA secondary structure or DNA secondary structure, it is always your hydrogen bonds. Okay, Tanvi. Now, so other regions of RNA molecules have no complement and hence are single stranded. So jahan pe if you see that there is no complementary sequence present, then they are going to be single stranded. And because of this single stranded and double stranded structures, you are going to get such kind of structures in your 2D, two-dimensional. Okay. Now, later on, what happens when they are in your 3D? So with the help of the X-ray crystallography, we came to know that there is a L-shaped structure which is formed from your this structure, which goes to the L shape formed of your tRNA molecules. And once the tRNA molecules are in this shape, then only they get charged with the amino acids. Okay. So tRNA molecules show a characteristics and highly conserved pattern of single-stranded, double-stranded regions, which is shown as clover leaf structure. The principal features of the tRNA clover leaf are, of course, the acceptor arm. Okay, then there is a 3-stem loop. So those are your pseudo-uridine, dihydrouridine, variable loop is there, and the other 4th loop, which is meant for the anticodon loop. Okay. Now, so these are the like characteristics loops. So let's talk about each of them. So the acceptor arm. So all these references has been taken from the Watson book. Okay. तो अगर कुछ डाउट आता है जहाँ पे कुछ समझ में नहीं आया, so you can just go back to the Watson reference book. I'm using 8th edition. You can just cross check with that. So acceptor arm is so named because it is the site where the amino acid is going to get attached, right? So यहाँ पे आपका amino acid आएगा. So that's why it is known as the acceptor arm. The 5-CCA sequence at the extreme 3- end of the molecule in a single-stranded region that protrudes from this double-strand stem. So ये हो गया आपका acceptor arm. Okay. Then coming to the pseudo-uridine loop, it is called because of the presence of the unusual bases जो भी हमने discuss किया. Okay. Now the modified base is often found within the sequence of T, pseudo-uridine, U, CG. Okay, I hope you're able to follow over this. Now, the D loop takes its name because of the presence of dihydrouridine in this group. Already हमने mention किया. Then anticodon loop is there, of course. So these anticodon means what? अगर मेरा AUG mRNA में कोड कर रहा है तो यहाँ पे क्या होना चाहिए? This is 5-3- sequence of mRNA AUG, right? तो यहाँ पे 5-CAU होगा, right? So anticodon में क्या present होगा? UAC. Agree? Yes, are you
Yes,
able to
ma'am.
follow? Yes, okay. So उसी तरह से आपका स्ट्रक्चर भी वैसे ही प्रेजेंट होगा. ओके, तो जैसे कि मैंने अभी यहाँ पे दिखाया 5-AUG है, दिस इज योर 5-3- ऐसे जा रहा है, राइट? तो यहाँ पे मुझे क्या चाहिए? तो इट हैज टू बी इन सच ए वे, ट्विस्टेड इन सच ए वे दैट इसको यहाँ पे बेस पेयरिंग करने के लिए 5- यहाँ होना चाहिए, 3- यहाँ होना चाहिए. ओके, then only they can form the base pairing. This is just to read, okay, this is a correct tRNA which has been brought by the enzyme, and then the amino acid is the correct one. Okay, so ये तो एक हो गया. This is like called the reading of the codons. So this is just the reading. The correct base pairing means here the reading is happening only. Okay, now what is happening? So we know that later in the ribosome we have got those EPA sites are there in the 50S subunit of the ribosomes where the actual protein synthesis takes place. We'll come to that later. Okay, the variable loop sits between the anticodon loop and the pseudouridine loop, and it, as its name implies, it varies in size from 3 to 21 bases. So guys, abhi bhi it's not confirmed like why this variable loop is present. कुछ लोगों ने बोला है that it helps in distinguishing between the tRNA synthetases. Okay, so it is helping the enzymes to bring the correct tRNA molecule so that it can get charged with the correct amino acids. So people have said that this is also one of the reasons. Now there is one scientist in not in at Delhi. I hope you have heard of it. ICGEB करके center है at Delhi. So there's this guy who is working on the mycobacteria. Okay. So अगर आपने कभी notice किया हो in the science news also, the north region of India basically work on the tuberculosis. Okay, and south region of India they basically work on, any idea किस पे काम करते होंगे? बेसिकली कैंसर पे तो पूरी दुनिया करती है. ओके, इंडिया में नॉर्थ रीजन माइकोबैक्टीरिया पे करता है ट्यूबरक्लोसिस, एंड साउथ रीजन में बेसिकली दे वर्क ऑन अनदर डिसीज. कैन यू गेस द नेम ऑफ द डिसीज?
Diabetes mellitus.
Correct, absolutely correct. Okay, what made you to say diabetes? क्यों लगा डायबिटीज ही है?
Ma'am, just guessed.
Acha, okay, no problem. So, but it was a correct guess. See, it's not like written anywhere, but like I came to know about this because I have attended many conferences, right? So I have been to north and south everywhere, and what I've noticed is that like even they have mentioned. So the regions where this what to say, the tuberculosis bacteria can grow. So we know that the tuberculosis bacteria, they can go to the dormant stage. If you know what is dormant, dormant means they can just go to the stationary phase where they're not growing, but they are alive, right? So inside the host, inside the humans also, inside the, they can just go infect the macrophages of the humans, and they can just stay alive inside that without getting killed. So macrophages are meant to remove these bacteria, right? But the dormant mycobacteria are like, you know, they can just go into the dormant stage, and where they can just stay inside the macrophages without getting killed. So जब उनको proper conditions मिलते हैं, host का अंदर सब बिगड़ जाता है, then they start, you know, replicating and causing the tuberculosis in the host system. So what I've seen is that the temperature, the climate, everything is very good for the TB bacteria to grow in the north region, and basically most of the institutes are working over there. Then the same, that heat or the temperature what I'm talking about is also seen in Chennai. Okay, so that's why Chennai is also a center for TB and the north region. This I'm talking about the summer summer season, okay, not the winter wala, in the summer season. Now, in the case of south region, I said diabetes because the main factor for diabetes is sugar, we all know, right? But north में तो आपका sugar cane होता है. But होता लोगों को किन लोगों को सबसे ज्यादा होता है? जिनका diet mainly consists of rice. So it's not that if you eat sugar you get diabetes. It is the glucose intake which is getting affected, right? So diabetes आपको मालूम है किस लिए होता है? When your insulin is not working properly, आपका insulin receptor काम नहीं कर रहा है या फिर आपका insulin produce नहीं हो रहा है. So these are the two ways by which you get diabetes. Okay, and the diet which is affecting is that the rice वाला. It's not like आप sugar cane जितना ज्यादा खाओगे या sugar खाओगे तो आपको वो हो जाएगा. But what we have seen is like the habit, the lifestyle people have adopted over period of time has developed into such problems. So जो South Indians हैं, जो mainly South region है, so those people, even though they are very thin, okay, they are very thin. They also work in the field for the, growing their paddy fields, etc. So they are also very hardworking. They are very thin, but the diet and the, the evolution or the natural selection, I should say, whatever it is, it has evolved in such a way that the people whose diet are rich on the rice, they get diabetes early. So 30-35 के बीच में आजकल हो जा रहा है सभी को. Now it has become very common. पहले common नहीं था. अभी it has become common unless until it is genetic. So this is because of the habit, because of the lifestyle, eating lifestyle. और genetic cases दिन तो आ ही जाते हैं. Okay, so that is just separate story. And all over the world, of course, it's the cancer. As I mentioned, there are so many types of cancer. And कैसा होता है ना, unless maybe I don't know, आपके nearby कितने लोग affected होंगे. I mean, it's good that people are not affected. But if you hear about like someone close or जो भी neighbor में हो या उनके किसी रिश्तेदार में, so the rate of cancer हमें कम दिखता है. Right, but once you're in that field, you will see that all over the world, the rate of cancer is really very, very high. And the main reason is all the bad lifestyle. Okay. And abhi aisa ho gaya, even if you know that this can cause cancer, you just can't go, you just can't leave that. I'll give you one example. In one of the conference, they have proved that eating McDonald's ka French fries and burger can cause cancer. Okay. But they have proved, they have shown the experimental data also. I have seen the data and I got convinced also. But the only thing is that once or twice in 6 months, even I go and eat. So, wohi ho jata hai na? Like, even if you know, but you don't change. Anyways, that is different story. So, if you guys can stop eating McDonald's ka French fries and burger, it's good. Okay. I'm not against them, but घर पे बनाओ and खाओ. I can just suggest that only. Anyways, coming back over here. So the tRNAs have L-shaped three-dimensional structure. So I already said that it is because in the crystallography, that X-ray crystallography, they have shown that it has got the L-shaped structure. Okay, so the acceptor arm, the terminus of the acceptor stem is at one end of the molecule and anticodon. So उनके बीच में 70 angstrom का distance है. Okay, I'll show the figure. So how they have shown is when from 2D to 3D jab jata hai. So this is all happening in the cytoplasm. So you can see that how nicely they actually overlap with each other and form the structure. So the distance between the acceptor arm, this one, and the anticodon, they say that it is 70 angstrom. Okay, ये एक important part है जो आपको याद रखना है. Because this is going to be used in your tRNA synthetases also. Okay. Okay, so I'll just repeat once again. So from the X-ray crystallography, we'll discuss about this technique also. It is important technique, a part of your methods in biology. So what they do is from the cell, they get all this tRNA molecules, they crystallize it. Okay, they make it small crystal, they diffract and they try to solve it. Okay, and over here also you need to know the sequence. So we all have the sequence of the tRNA molecules. Then you solve the structure. So what they have seen is that in the crystal structure, after solving the crystal structure, they found that the molecule is in this shape. Okay, and that's why they have said that it is L-shaped molecule. So tRNA molecule exists in L shape. Now this transformation from your, this is 2-dimensional, this is 3-dimensional. Okay, so this transition from 2D to 3D, it is happening in the cytoplasm. So if you see that these loops are getting twisted little, or you can say that overlap. Can you see it over here? This is your anticodon. Anticodon आपका move नहीं हो रहा है. Okay, rest all is moving. This portion has got straightened up. ये आपका इस तरफ चला गया, and this has come over here, this direction. Can you see? Right? So this has resulted in this particular structure. Now, so this is made it like, so the acceptor arm is there and the anticodon over here. So the distance between the anticodon and the acceptor arm, okay, this distance is 70 angstrom. This is important to remember. Now, if you see that your pseudouridine loop and dihydrouridine loop is exposed outside, so these are going to help in selection of your tRNA synthetases. Okay. Now, these tRNA synthetases will, with the help of D loop, pseudouridine loop, and the anticodon, will make sure that this particular tRNA is getting charged with the correct amino acid. Okay, is this much clear?
Yes, ma'am.
Okay. Now, the attachment of amino acids to tRNA. So this is happening in 2 steps. Okay. So they are attached, sorry, they are charged with the attachment of amino acids to the 3' terminal adenosine, as we have discussed just now, that 5-CCA3', right? So आपके जो end में है वो हमेशा A रहेगा. To this, your amino acid is getting attached, and the bond is your acyl linkage. And then later on, it is your peptide bond. Okay. Now, the tRNA molecules to which amino acid is attached, it is called charged tRNA. If there is no amino acid, then it is said to be uncharged tRNA. Simple. Now, charging requires acyl linkage between the carboxy group of the amino acid. Please remember, okay, it is a carboxy group of the amino acid and the 2' or the 3' hydroxy group of the adenosine nucleotide. So as I mentioned, right, either it will be 2' OH or 3' OH of the ribose of the adenosine, okay, which is present. ये आपका A का 2' OH होगा या फिर 3' OH होगा, depending on the tRNA synthetases class. So this acyl linkage, it is a high energy bond because its hydrolysis results in large change in free energy. Now, to do this thing, always remember synthetases. जहां पे भी synthetases word आता है, it always uses ATP. Okay, अगर मैं synthase बोलूं, then it produces ATP. Getting the point? Able to follow?
Yes.
हाँ, so एक बार और बोलती हूँ. Synthetases अगर आता है, then it is using ATP. If it is synthase, it is producing ATP. So ATP synthase आपने सुना होगा, correct? So those are producing the ATP molecule. Now this acyl linkage, it's your high energy bond. Now this high energy is required because I'm going for protein synthesis. I need energy for the bond formation. Now we'll see how these bonds are formed. So this is significant for protein synthesis. The energy released when this acyl bond is broken is coupled to the formation of the peptide bond. Now acyl bond कब break होगा? From the tRNA molecule, from the charged tRNA molecule, the amino acids will be taken to the peptidyl transferase. And when the bond is broken between the tRNA and amino acid, that energy is taken to create peptide bond at the ribosome P subunit. Okay, we'll come to that, don't worry. Okay, is this slide is clear? Okay, we haven't discussed much over here, but the next slide is important. Okay, this is just the representation where you have got the acceptor arm and then the anticodon regions are there. So same thing, You can see the color coding over here, like kaise purple area is present over here, the green and the red and the blue regions over here present. So 2D model to the 3D model, just the representation, okay? And you can see that each nucleotide bases are inside of the structure except your A, which is present outside. Can you see? So we also know that this is single stranded. This CCA hamara tha, right? So it is single stranded, and you can see that this A is protruding out, and which is required so that the amino acid can come and get charge over there. Okay. Now, so amino-acyl tRNA synthetases, they are the enzymes which charge your tRNAs in the two steps reaction. Okay. So when we say two-step reaction, but everything is happening at the same time on to the enzyme. Okay. Now let's see what are those steps, and later we can see how the enzymes are actually carrying out these steps. Okay. So first you have got the amino acid. We haven't mentioned which amino acid. Okay. So it is kept as R group. Then you see that your amino acid is getting first the ATP, the charge. It is getting charge, okay? So when you say charge amino acid, it means what? It is going to get adenylated. Since it is ATP, so you can see that the carboxyl group of the amino acid is attacking at the first gamma phosphate group. Can you see? Okay. So what is being released then? Your pyrophosphate. और हमने फर्स्ट लेक्चर में ही डिस्कस किया था when the pyrophosphate with the help of the phosphatase, okay, with the help of the phosphatase it converts your pyrophosphate into inorganic phosphate, right? So it is breaking the bond between these two phosphate, and that generates enormous amount of energy. Okay, so that energy is released into the system, which makes your delta G negative and making the entire reaction spontaneous. Okay. Now, next step. So what is happening? This adenylated amino acid. So obviously each amino acid is going to be adenylated amino acid. Okay. Let it be any other glycine ho, serine ho, threonine ho. Every amino acid before getting charge onto the tRNA is going to form the adenylated form of the amino acid. Now, again, so this 3-OH, for example, okay, we are just talking about the 3-OH. आपका ये 2-OH भी हो सकता है, depending on the class of the tRNA synthetases. Okay, so depending on that, this 3-OH is going to attack at the carboxyl group. So what will happen? Now this your acyl linkage is formed over here between the tRNA and the amino acid. This is called your charged tRNA. Finally, the AMP is released into the cytoplasm. Okay, are we cool? Clear? Yes, Tanvi, कुछ doubt है? Able to follow?
Ma'am, maybe repeat the second step.
Huh.
B1.
Okay, no problem. So what happens over here is that, see, so you have got 20 amino acids. So each 20 amino acid first will be in the adenylated form. Okay, so this is clear that it attacks over here. Your pyrophosphate is released with the help of the phosphatase. This will become 2-PI, that is your inorganic phosphate, and this generation release so much amount of your energy. Okay, now second case, what happens is that the tRNA. So depending on the classes of the synthetases, there are two classes, 1 and 2. Okay, whether it is 2-OH or 3-OH, depending on that, your tRNA molecule 3-OH is going to attack at the carboxyl group. Clear? So what will happen? This OH and COOH over here is there. So what will happen? ये AMP के phosphate को यहाँ पे आपका O negative छोड़ देगा, यहाँ पे वो bond बना लेगा. ठीक है? Okay. So what will happen? This bond is formed. Can you see? COOH bond is formed over here. Clear?
Yes, ma'am.
Okay. So AMP is finally released into the cytoplasm. Now you have got the charged tRNA. So this is ready now to go to the ribosome and mRNA where it is present and start giving your amino acids to the required codon as per the required codon. Okay, so these are the two steps which is carried out by the tRNA synthetases, amino acyl tRNA synthetases to charge your tRNA. Okay, so first is the adenylation of your amino acid. So how do you say? First is the adenylation of your amino acid, and then second is the charging of tRNA. Okay, so charging of tRNA means आपका 3-OH is going to attack on the carboxyl group of the amino acids, forming acyl linkage over there, releasing AMP into the system. Clear? Okay. Now look at this figure. So let us imagine that this is our amino acyl tRNA synthetases for methionine. Okay, so first your enzyme is going to recognize your tRNA depending on the pseudouridine, dihydrouridine and the anticodons. Okay, ये हम discuss करेंगे in detail how they help in the recognition of the amino acid. Okay, so for right now just remember, based on the pseudouridine and dihydrouridine anticodons, the tRNA synthetase will understand that this particular tRNA should be charged with methionine. So this tRNA will come, it has got the ATP binding pocket also, so we know that, and methionine यहाँ पर आएगा. So methionine first gets adenylated. Okay, it is going to release your 2-PI. Later on what will happen? This adenylated methionine will get charged onto over here. Okay, releasing your AMP later. So at the end what is happening, even though it's a two-step reaction, but in the enzymes it is happening at the same time. Okay, so first adenylation of amino acid is happening, and that adenylated amino acid is getting charged on the tRNA molecule. So this is how each tRNA molecule is getting charged with the respective amino acids. Clear this figure? Yeah.
Yes, ma'am.
Okay. So this is the crystal structure. So this is how, so what they have done is they have showed the enzyme in this model, and you can see the ball and stick model of the tRNA over here. So you can see how nicely the anticodon region is present over here, is being used by the enzyme, right? Then it has got the catalytic binding pocket जहाँ पे ये काम हो रहा है, and the editing pocket. So this is your amino acyl tRNA synthetase. So editing pocket, catalytic pocket. So what is happening? The amino acids as well as your ATP comes and binds, and finally it is loading the adenylated amino acid on the acceptor arm of your tRNA molecule. Okay. अगर आप structure को ध्यान से देखो, so you can see that the acceptor arm जो है आपका amino acyl tRNA synthetase के अंदर की तरफ है, and the anticodon also, whereas the rest of the regions, okay, are towards the outside. But the arrangement or the alignment of the tRNA molecule in such a way that it is being recognized by the enzyme for the correct charging of the amino acid. Okay. Yes, able to follow. थोड़ा सा है complicated, but yeah, I'll try to get video of this if I could get, and maybe in the next class I can show. So I hope this much is clear. So इतना भी ध्यान में रख लो that amino acyl tRNA synthetase is there. First, it is charging your amino acid, and that charged amino acid, that adenylated amino acid is then finally charged onto the tRNA molecule at the acceptor arm. ठीक है? ये सारा याद रहेगा. And please always remember 5-CCA है आपका sequence for each tRNA molecule. It is same. This is conserved. Okay. So this much you can remember. Now, what are the two different classes of amino acyl tRNA synthetases? So here I am giving you the list, and this is directly from the textbook itself. So you have got the class 2 and the class 1 molecules. So you can see class 2 में क्या-क्या आते हैं? Glycine, alanine, proline, serine, threonine, histidine, aspartic, asparagine, lysine, phenyl, and rest in your class 1. अब इन दोनों में अंतर क्या है? So class 1 enzymes, they are which attach the amino acids to -OH of the tRNA molecules, and they are monomeric in nature. तो इसका मतलब क्या? The tRNA amino acyl tRNA synthetases are monomers only. Okay, whereas in the case of class 2, they attach amino acid to the 3-OH of the tRNA molecules, and typically they are dimers or tetramers. So you can see that the size or the structure of the tRNA synthetases are also different. Okay. So आपको बस ये थोड़ा सा और याद रखना होगा. It is simple also, and you can see it over here. So basically class 1, if you see the quaternary structure, they are monomers. सिर्फ एक दो cases होंगे जहाँ पे they are dimers, but rest all mainly they are monomers. Okay, so they are monomers over here. So either you can remember this part and rest will come to the second class, or you can remember this. So ये आपके ऊपर आप कैसे इसका acronym बना सकते हो or whatever. Just remember one of the classes so that the rest is falling in the next class. So you can remember this, like maybe you know the negative charge or the positive charge are coming over here, something like that. I don't know. मैंने तो पूरा रट्टा ही मारा था, but now I don't remember. But it is on you now how you can remember. Okay. So the class 1, class 2 main differences. Class 1 is where the amino acid is attached with the 2-OH of the tRNA molecules, and class 2 हो गया जहाँ पे आपका 3- में आपका attach हो रहा है. ठीक है. Okay. This is clear, I believe. This is very simple. Now, I think this is the same thing. So the tRNA synthetases, when recognized in charge more than one tRNA, it is known as iso-accepting tRNAs. Okay, this is what I mentioned in the starting of the lecture, iso-accepting tRNAs. Nevertheless, the same tRNA synthetase is responsible for charging all tRNAs for a particular amino acid. Thus, one and only one tRNA synthetase attaches each amino acid to all of the appropriate tRNAs, and so most of the organism will have 20 different tRNA synthetases instead of having 61. Okay, but this is not always the case. So we have got certain peculiar scenario where there are only one synthetase which is charging all the tRNA molecules. Okay, for example, some bacteria, they lack synthetase for charging tRNA for glutamine with its cognate amino acid. So what we mean by cognate amino acid, यानी कि this tRNA should be charged with its glutamine, right? तो वो उसका cognate amino acid हो गया. Instead, a single species of amino acid tRNA synthetases charges this tRNA glutamine as well as tRNA glutamate. Okay. अब दोनों में अंतर क्या है? If you see the structures of glutamate and glutamine, what is the difference? Can you tell me? I have always mentioned that you need to know the structures of the amino acids. कभी ना कभी कहीं ना कहीं ये लगेगा ही लगेगा. So if you see the structures of glutamine, what is present? Glutamine नाम से ही पता चलना चाहिए.
It has an extra amine group, and glutamate has a carboxyl group at the end.
Yes, very good, very good. So now keeping that thing in the mind, so the second enzyme then converts by amination the glutamate moiety of the charged tRNA glutamine molecules to glutamine. ठीक है, तो उसने क्या किया है, उसको पहले the tRNA glutamine वाला जो आपका tRNA है उसमें पहले glutamate डाल दिया है, उस glutamate को later they have converted to the glutamine. Okay, so this glutamate glutamine is aminated to then glutamine tRNA glutamine. I hope this is clear. Should I repeat? थोड़ा सा confusion हो जाता है. So in short, what I'm saying over here is that in few bacteria, you don't have glutamine tRNA. Okay. What they do is they have got glutamate ka tRNA molecule. So they are going to charge the glutamine tRNA also with glutamate. Okay, then the another enzyme will come and convert this glutamate to glutamine. So overall we have got only a single amino acid tRNA synthetase, but additional enzyme is coming which is converting glutamate to glutamine. अभी clear है? Yes, Akshaya?
Yes, ma'am.
Huh? I hope it is clear, and see, I just want that it should be clear because एक तो exam में ये नहीं आएगा. Don't worry about it. but we are I just want to make sure that these informations are given to you. ऐसा नहीं होना चाहिए कि that it is universal कि methionine के लिए methionine ही है, glycine के लिए glycine ही है. There are cases where you have got different amino acid charging also, but then second enzyme is coming and converting that particular amino acid to the next amino acid. Okay, so there are cases available. This is not universal. Okay. Wow, thanks. So I'll be stopping here, guys, now.